Okay, thank you for the opportunity to speak at the workshop. My name is Rashmie Abeysinghe, and the title of our paper is Auditing Hierarchical Relations in the Pharmaceutical Biologic Product Subhierarchy of SNOMED CT. My co-authors are Dr. Gong and Dr. Licong. So this is a brief outline of my presentation. So first, I will introduce you to SNOMED CT, which is our focus here, and then I will explain why ontology quality assurance is needed, and then I will in detail discuss the method and show you some of the results, and also talk about some interesting future directions. So SNOMED CT, of course, is very popular. It is the largest and most comprehensive clinical terminology system in the world. It contains medical terms providing codes, terms, synonyms, and definitions to be used in clinical documentation and reporting. And it also provides the core terminology for electronic health records. So the current version, or the version that was used in this work, which was the March 2021 US edition, contained over 360,000 concepts, which were hierarchically arranged in 19 different subhierarchies, like clinical finding, body structure, procedure, and pharmaceutical biologic product, which was actually our focus in this work. So pharmaceutical biologic product actually contains around 20,000 concepts, and which also covers a wide range of drug related products. So biomedical ontologies like SNOMED CT, they're generally incomplete, underspecified, and non-static, and they are constantly evolved or changed to reflect the state-of-the-art knowledge of the domain that they model. So during this process, errors or inconsistencies may be introduced, even though how much care is taken to avoid introducing inconsistencies. Because these ontologies have become very large and complex, so it is in some cases it's unavoidable that errors would be introduced. So there could be different types of errors, like missing relations, erroneous existing relations, redundant relations, or like missing concepts, and so on. So the issue is that these errors could actually propagate to downstream applications that use these ontologies as knowledge sources and make them inaccurate as well. So for instance, this figure actually denotes missing is a relation or missing hierarchical relations in the clinical findings subhierarchy of SNOMED CT. So the red arrow is actually a missing ISA. So for a task like clinical cohort identification, ontology-based clinical cohort identification, where you need to identify a patient cohort having this disease, malignant epithelial neoplasm of skin, then you're going to miss patients with this disease, squamous cell carcinoma of skin, because we don't have this link in SNOMED CT. So because of such issues in tasks that use these ontologies, we have to identify these issues and fix them. It is essential. So ontology quality assurance has become an essential part in ontology management life cycle. However, manual review is not really sustainable because how large and complex these terminologies are, and it could be very time consuming and also labor intensive if you try to manually audit them. So automated methods are actually very highly desirable to audit biomedical ontologies. So now I will discuss the method that we undertook to audit the pharmaceutical biologic product subhierarchy of SNOMED. So we basically we had three major steps. So we first extract ingredients from concept names in this subhierarchy, and then we generate replacement candidates for these ingredients, and then we replace these ingredients with the replacement candidates to form superconcepts. So this is how we actually identify ingredients from concept names. And we noted that majority of concepts in the pharmaceutical biologic product subhierarchy exhibited either of these two lexical patterns. So the first one is prefix followed by an ingredient, and the second one is prefix followed by ingredient, which was followed by the word in and endorsed form. So we focus on concept in this work. We focus on concepts with prefixes product containing only and product containing, and we saw that this actually covers around 93% of concepts in this particular subhierarchy. So for example, for a concept with pattern 1, this is how we extract its ingredients. So we first, we simply obtain the entire substring after the prefix. So for example, for the concept product containing carbonic anhydrase inhibitor, so the prefix would be product containing, and we simply obtain the entire substring, which is carbonic anhydrase inhibitor as the ingredient. For pattern 2 with prefix, ingredient, and a dose form, it's slightly different. So we obtain the substring between the prefix product containing and before the word in, which means azithromycin is the ingredient of the concept product containing azithromycin in ocular dose form. So this is how we obtain ingredient names from concepts. And generating replacement candidates for ingredients. So what we do here is we check whether an extracted ingredient exists as a SNOMED CT concept itself. So by that, what I mean is whether there exists as another SNOMED CT concept with the same name as the extracted ingredient. In such cases, actually, this SNOMED CT concept would almost always be belonging to a separate subhierarchy other than the pharmaceutical biologic product subhierarchy, something like the substance subhierarchy. Yeah, and we identify all its ancestors, which are parents, grandparents, and so on. And these would serve as replacement candidates for the ingredient. So for instance, for the concept product containing aminotransferase inhibitor, the ingredient extracted would be aminotransferase inhibitor. And we found that this actually forms the SNOMED CT concept itself. So we identify its ancestors, which are transferase inhibitor, enzyme inhibitor, effector, and disposition as replacement candidates for this ingredient. So the next step is to identify super concepts. So here what we do is we generate a set of potential super concepts by iteratively replacing the ingredients that we extracted by their replacement candidates. So for pattern 1, we hold the prefix or leave the prefix as it is and replace the ingredient name with the replacement candidates. So if we have K replacement candidates, we would have K different potential super concepts. It's similar for pattern 2 as well. So we hold or leave the prefix as it is and also leave the word in and dose form as it is and replace the ingredient with the replacement candidates. So if a potential super concept identified generated this way exists as a SNOMED CT concept, and there is no existing ESA relation between the original concept and this potential super concept, then we say that this is a missing relation in consistency. So for instance, if you consider the same example, product containing aminotransferase inhibitor with aminotransferase inhibitor as the ingredient and these 4 replacement candidates, we identify these 4 potential superconcepts by replacing, by keeping product containing the prefix as it is and replacing the ingredient aminotransferase inhibitor with iteratively with each of these replacement candidates. So replacing with transferase inhibitor, we get product containing transferase inhibitor. Replacing with enzyme inhibitor, we will get product containing enzyme inhibitor, and so on. However, product containing disposition actually does not form a SNOMED CT concept. So that means there does not exist any other SNOMED CT concept with the name product containing disposition. Same for product containing effector. It does not form a SNOMED CT concept. Product containing transferase inhibitor forms a SNOMED CT concept, but it is already a parent of A. So we actually ignore these three cases. However, product containing enzyme inhibitor is a SNOMED CT concept, but it is not an ancestor of A. So in this case, we say that this is a missing relation. So there should be a missing relation between product containing aminotransferase inhibitor and a product containing enzyme inhibitor. So we apply this method to the pharmaceutical biologic product subhierarchy of the March 2021 US release of SNOMED. And we identified 87 potential missing ESA relations. And a domain expert evaluated each of these 87 cases to check how many of them are valid. And it was revealed that 85 of them are actually valid cases of missing ESA relations. So these are some examples of the missing ESA relations identified out of the 85 cases. So for instance, product containing arnica extract should be a subtype of product containing plant agent, and so on. So in conclusion, we applied a lexical pattern-based approach to identify missing ESA relations in the pharmaceutical biologic product subhierarchy of SNOMED CT, where we identified ingredients from concept names using particular lexical patterns, and then identify replacement candidates for those ingredients, and then identified potential superconcepts for those original concepts. So something that we would like to do in the future is to experiment with synonyms, because for this work, we did not consider synonyms. We only considered the fully specified names of SNOMED CT concepts. So if we had considered synonyms, we believe we would have gotten maybe additional missing ESA relations. And also for this work, we identified ingredients using a lexical pattern from the concept name. But while this covered the majority of concepts in this particular subhierarchy, some concepts did not fall into this lexical pattern. However, we can find the ingredient information among the logical definitions of the concepts, which we would like to look into. Also, we would like to explore the possibility of applying this method to other biomedical ontologies. So in this case, actually, we would have to explore new lexical patterns, because this particular lexical pattern was crafted specifically for this particular subhierarchy. So we would have to identify such cases, such lexical patterns that will work for other maybe subhierarchies of SNOMED or other biomedical ontologies. So this task actually could be a bit of time consuming. So we'd like to see whether we could generate these lexical patterns automatically by leveraging the existing relations of each ontology, which is something that we are currently looking at. So that is the end of my talk. I would like to thank, we'd like to thank NIH for their funding support. Yeah, thank you.
Thank you, Rashmie. Do we have any questions from the audience from Rashmie? Yes, Asha?
Yes, Rashmie, if you find out those missing relations, do you have a channel to feedback to the SNOMED? I think they will
yeah,
actually add it.
Yeah, that's something really that we really are looking forward to. So in our previous work, actually, we did some quality assurance work for NCI, where we work with NCI EVS, who are the curators of NCI to add those missing relations that were found to the NCI. So likewise, we will communicate with SNOMED to have these missing user relations added to SNOMED, maybe in a new version, new release.
Yeah, I also had a suggestion to the organizers. Like I feel in Xinjuan's work will be very interesting to Medra, because I know them. I think they will. If we invite them to this workshop, they will make time to listen to this talk, and so as to SNOMED CT people, I would say. So in the future, yeah, I can help you if you need to bridge those inviting key stakeholders.
That's a good suggestion. Thanks, Asha.
Welcome.
Okay, any other questions?
Yeah, I have one question. Yeah, or maybe one comment. I think you suggested in the discussion by exploring the logical definition, right? So because when I look at your examples, I feel like if you have logical definition in the certain, some of the missing like iteration actually can be inferred automatically. Like it's a product containing some enzyme, right? And then, so if at the protein level you already assign the protein is an enzyme, right? Then like automatically it can be inferred somehow, magically, the missing parent, right? So the product containing enzyme. So I think, somehow similar. There are many cases you can probably explore the usage of the logical definition for automatically inferring the parent. In herbal fungi field, actually, we prefer to not asserting multiple inheritance. We prefer have a single inheritance, but many other can be inferred, right? So it actually can support like a maintenance of the ontology.
Mm-hmm. I think the cause of these missing user relations are due to like incomplete logical definitions, actually. So if the logical definitions are complete, or they are like fully defined concepts, these missing user relations may be, as you said, automatically inferred through a reasoner. But there are some cases where in these cases where the logical definitions are not complete. So that's why we have these missing cases. So SNOMED actually also releases an inferred version, which a reason has already been run. Yeah.
Okay, thank you. Yeah.
Rashmie, how many actually missing such relations do you expect to do you think you have in this in the branch, and where you identified 85, right? How many do you expect? And have you considered using other approaches, anything available, like in terms of benchmarking to compare the performance?
Okay, so for the first question, I think it really depends on the particular subhierarchy. So for other subhierarchies, for this one, it contained around 20,000 concepts. For like clinical finding, it has around like I think more than 100,000 concepts. Mm-hmm. So it would of course contain many more errors, and most of the concepts in this particular hierarchy were actually fully defined. So we expect less errors in this subhierarchy than maybe a subhierarchy like clinical finding, where you have like a lot of primitive concepts. And for the second question, actually, benchmarking is a difficult thing to do because like ontology culture students, these approaches are like discovery oriented. So what we find is like previously unfound errors. So different approaches find different kinds of errors. So that's why we have to rely on a domain expert. So because, I mean, simply one method having like high performance does not mean that it is better than another approach with a lower performance, simply because they might be finding different kinds of errors. So yeah, it's difficult to compare.
Okay, thank you.